It is now my pleasure to introduce our presenters for today. First, we will have Mary Nilsson presenting on common mistakes when pooling clinical trial safety data. Mary Nilsson is a statistician at Eli Lilly and Company. She has worked as a clinical trial researcher for over 34 years with an emphasis on clinical trial safety analyses. Additionally, she co-leads the PHUSE Safety Analytics Working Group, an FDA PHUSE public-private partnership involved with creating cross-functional education and cross-industry recommendations for standard safety analyses and displays. Next, Dr. Veronica Pei will be presenting on FDA Type C meetings on integrated summary of safety or ISS safety analysis strategy and related data requirements. Dr. Pei is a board-certified emergency physician and a commissioned officer in the U.S. Public Health Service, currently serving as Associate Director acting for the Bioinformatics team in the Office of New Drugs. Dr. Pei is involved in the development, implementation, and support of bioinformatics initiatives, as well as data standards within the Office of New Drugs. Now, please join me in welcoming our first presenter, Mary Nilsson.
Hi, thank you for that introduction. Veronica and I are happy to be here to talk about clinical trial safety analyses. Since this webinar offers continuing education credits, we do need to have some learning objectives, and these learning objectives were provided in the announcement for the webinar, but we'll review them here real quick. So our objectives are to understand when to contact the FDA for discussion around integrated safety analysis plans, understanding common mistakes in integrated safety analysis, and using best practices for integrated safety analyses. As noted in my introduction, I am a statistician at Eli Lilly and Company working in a safety analytics group. My primary role is to consult with submission teams on their analysis plans for integrated submission documents, such as the Summary of Clinical Safety or the Integrated Summary of Safety. I'm also a co-lead of the PHUSE Safety Analytics Working Group, which is part of a public-private partnership with the FDA. So today, I'll be presenting on behalf of the PHUSE Safety Analytics Working Group. We'll start with a couple disclaimers. This first disclaimer is the standard disclaimer that we use in all deliverables from the FDA PHUSE partnership. It basically says, the opinions expressed are those of the author and should not be construed to represent the opinions of any PHUSE members or their companies or organizations or any FDA views or policies. I added this second disclaimer to be transparent that the examples included in this talk are not necessarily from actual submissions to the FDA. They're from the collective experience by members of the PHUSE group, and some might be seen during planning and addressed prior to submission. First, I'll provide a little background on PHUSE and the FDA-PHUSE collaboration. PHUSE is an independent, not-for-profit organization that started in 2004. PHUSE is not an acronym for anything. It's used to stand for Pharmaceutical User Software Exchange, but it was rebranded when the scope of the PHUSE activities became broader. For more information, you can go to the PHUSE Global website, which is the main site that we use for PHUSE. There's also a working group tab that leads you to the current working groups and associated projects. There's also a manuscript published by FDA representatives describing the initiative, and here's the reference on the slide. Today, there are 8 working groups, with safety analytics being one of them. There are several projects within each working group, so across all the working groups, there are over 40 ongoing projects. So certainly encourage you to peruse the content on the site and familiarize yourself with some of the activities going on as part of this collaboration. So today's webinar is coming from the Safety Analytics Education Project team, which is part of the Safety Analytics Working Group. The Safety Analytics Working Group has the goal to improve both the content and the implementation of clinical trial safety analyses. I co-lead this working group with Greg Ball and Scott Prostel, and the education project team is led by Bill Palo and Chris Smith. While the education project team is relatively new, other safety-related project teams as part of the collaboration have already produced many safety-related deliverables over the past 10 plus years, and several of these deliverables are included in the reference section at the end. So hopefully the background is helpful in understanding why we are having this webinar. In order to improve both the content and implementation of clinical trial safety analyses, we all need to improve our safety analytics expertise. In particular, there's a significant gap in the knowledge needed to conduct proper integrated analyses. The gap spans multiple disciplines, including statistics. There's a myth that safety analysis, planning, and interpretation is easy, and it's just not. Also, there are some aspects of predominant practice that can be improved. It's difficult to change predominant practice without improving this expertise. Additionally, there's a general lack of awareness of useful resources. A lot of useful information is already out there, but not everyone knows about them or knows how to find them. Of course, we'll always have analytical challenge, which requires additional research. Safety analytics is an evolving field with all sorts of challenges that need some really smart people to help solve. For the remainder of this talk, I'll go over some common mistakes with respect to integrated analysis planning for submissions. It's not an exhaustive list by any means, but hopefully it highlights some key concepts submission teams need to know. So one common mistake is to not have an integrated planning document or not having one early enough. Most sponsors are likely doing a good job creating statistical analysis plans for individual studies. However, we don't think it's always routine across sponsor companies to create an analysis plan for integrated submission documents. When they are created, they're not always created early enough. For integrated safety analysis planning, industry practices vary widely. Some sponsor companies might produce an aggregate safety analysis plan, which has been introduced in the literature relatively recently. Some might produce a program safety analysis plan, which was introduced in the literature a little over 10 years ago. Others may produce an integrated safety analysis plan, which is a logical extension of having a statistical analysis plan for an individual study. However, some sponsor companies may not produce any textual analysis plan document at all and only create a list of analyses to facilitate programming. So let's take a moment to go over the differences between these document types. So statistical analysis plans describe the planned analyses, both efficacy and safety, for an individual study. The content is usually focused on what is planned for the clinical study report, but perhaps some companies include analyses for other types of deliverables, such as key manuscripts. Integrated safety analysis plans describes the planned safety analyses for the integrated safety document for a particular submission, such as the summary of clinical safety or integrated summary of safety. Perhaps some companies give such a document a different name. ISAP, Integrated Safety Analysis Plan, isn't necessarily a well-established term. Program safety analysis plans are conceptually broader than ISAPs. It's a molecule-level planning document. It would include the planned analyses for individual studies and integrated submission documents and may include additional information such as data collection decisions. Aggregate safety assessment plans are conceptually broader yet. It would include everything a PSAP would include, but can also include more detail around ongoing safety reviews and perhaps methods and processes around the IND safety reporting rule. It's currently unclear how many companies are creating PSAPs or ASAPs. Also, what is actually included in a PSAP or ASAP likely varies across companies as well. The scope depends on what other processes and procedures cover. Even within a company, there could be situations where one document type works better than another for various reasons. The pros and cons of these choices is out of scope for this talk, but perhaps will be covered in a future webinar. So let's pause and take a quick poll. This particular question is applicable to industry representatives. So if you're from a contract group that doesn't have their own procedures or from the FDA, you can take a sip of your coffee for a minute. So for those who work in industry, could you please help us out with this poll question? Does your company organization have a procedure outlining requirements for integrated safety analysis plans for the summary of clinical safety or integrated summary of safety in a drug application? Yes, no, or you're not sure. Okay, maybe just another few seconds. All right. As I mentioned, the pros and cons for the planning document type is out of scope for this talk, but we do recommend that sponsor companies at least consider creating a procedure with respect to integrated analysis plans if one doesn't already exist. The procedure would outline timing, ownership, and document type expectations. And the document type could remain flexible in a procedure. It's just good scientific practice to have documentation on plan analyses and when and why changes to plan analyses are made with rationale. I understand that perhaps this documentation is more important for individual studies with respect to what was planned before versus after any kind of treatment unblinding. However, there are still benefits in the integrated analysis space to have clear plans. Regarding timing, it probably goes without saying that early planning is better than later planning. Ideally, it would be great to have some documented molecule level planning in place before phase 2 studies begin. However, extensive planning is likely unrealistic at such an early stage. Prior to phase 3 studies beginning is a bit more realistic, but can also be challenging. Practices could be set up such that some aspects of planning can come earlier than other aspects. It seems to make sense to obtain alignment from all the right people on the data collection plan for safety topics of interest prior to protocol approvals to ensure the needed collection is obtained. Analysis planning would need to be in place, ideally in time for getting regulatory feedback and in time for programming and perhaps multiple table figure review iterations. So when you work backwards from a target submission date, that could be a year or more in advance of a planned submission. Regarding ownership, who owns what could vary on the size of the company, what functions the company has, and the scope of the planning document. Usually, analysis planning is owned by the statistics function. Regardless of ownership, integrated analysis planning should be viewed as a cross-disciplinary effort. Multiple functions, statistics, medical, regulatory writers should be involved. Regarding the document type, a procedure can allow flexibility. There could be different situations in which one document type is preferred over another for various reasons. Let's move on to another common mistake. Not asking for FDA input early enough. We recommend that you leverage FDA's offer to conduct a Type C meeting specifically on integrated safety planning. If that's not feasible for some reason, the integrated safety planning document can be sent to the FDA as part of another interaction, bundled with other questions. Veronica will cover more detail in her talk. Another common challenge is not understanding how to create integrated analysis sets. For this talk, I'm not making any distinction between the use of the word integrated versus pooled. Some companies may refer to these as pooled analyses or pooled analysis sets. I'm referring to defining how different studies and treatment arms are combined for purposes of an integrated submission document. For many submissions, not all pooling data from multiple studies is warranted and very helpful. When it is warranted and helpful, team members don't always come with the necessary knowledge to know how best to create these integrated analysis sets. When making decisions on how to pool studies, there are likely multiple ways that could be considered appropriate. However, there are some ways that might be considered wrong or clearly less preferred than other ways. In practice, there tends to be a lot of differing views on what should versus should not be pooled. Some researchers are very hesitant to pool anything, except perhaps studies that have an identical design, identical inclusion-exclusion criteria, and conducted from the same regions of the world. Other research might be very comfortable pooling studies across varying populations and designs. These decisions can take a fair amount of time to achieve alignment. Once you finally get alignment from within your team, it's yet another step to achieve alignment with regulators. When making decisions on what studies and treatment arms to pool, one important concept to understand is confounding. By definition, a confounding factor is a variable that influences both the potential cause and the potential effect. In the context of integrated clinical trial safety analyses, we try to avoid confounding of study and treatment. So let's consider this example in which a placebo-controlled study, study 1, is pooled with an active controlled study, study 2. And percentages are calculated simply by adding the little n's and the big N's within each treatment arm. We'll call this crudely pooled percentages. So in this example, you can see that the event of interest was a little less frequent in the placebo-controlled study than it was in the active controlled study. This situation is actually realistic because it's not unusual that the inclusion criteria is a bit more restrictive in a placebo-controlled study than an active controlled study. In other words, the patients in the active controlled study could have more advanced disease. And as you can see in this example, the crudely pooled percentages distort the results. There was no difference in the percentages between treatment arms in either study, yet the crudely pooled percentages would indicate a difference. When you compare the crudely pooled percentages across treatment arms, and the treatment arms come from different studies, you're not just comparing treatment arms. You're also comparing differences between studies. So study and treatment are confounded. So any differences or lack of a difference could be due to treatment, or they could be due to study. So in this example, the differences are due to study, distorting what you're really truly interested in, which is the treatment differences. You might be saying, why would anyone pool a placebo-controlled study with an active controlled study and provide crudely pooled percentages? Perhaps most researchers would know to avoid that, and we don't see it in practice as much as we used to. Early in my career, it wasn't all that unusual to see. However, what we do commonly see today is the temptation to pool placebo-controlled studies that have different dose arms. Then create crudely pooled percentages simply by adding the little n's and big N's across studies, and then try to compare dose arms. This analytical approach should also be avoided. Study and treatment dose are confounded. So as with the last example, when you compare the crudely pooled percentages across treatment arms and the treatment arms come from different studies, you're not just comparing treatment arms, you're also comparing differences between studies. You might be thinking, well, in this example, it's unlikely there are any differences between the studies. Perhaps, but it is an unnecessary leap of faith. Even if the inclusion and exclusion criteria are identical, different studies usually include different sites and perhaps different regions. So do you really want to assume there are no study differences? So here are some general recommendations when creating integrated analysis sets. We suggest ignoring phase designation. Pooling decisions for signal detection purposes are usually best made by looking at the actual study population, the doses, and the designs. Of course, it may turn out that it's only the phase 3 studies that have designs suited for pooling, but we think it's okay that a phase 2 study is pooled with a phase 3 studies if its population, doses, and design are suited for pooling. We don't think it should be excluded from a pool simply because it's designated a phase 2 study. Now, some regional labels may still choose to only include data from phase 3 studies, but that would be a separate conversation. There's a general alignment that healthy volunteers should not be combined with patients. Data from healthy volunteers and patients are usually presented in separate sections in a submission. Typically, creating an integrated analysis set that includes all patients ever on study drug is helpful. The display would include counts of patients having the event regardless of study, dose, design, just for the study drug patients. This integrated analysis set would have little value in assessing differences in treatment arms, but does have some value in finding rare events that might require further scrutiny through case reviews. Now, this might sound contradictory to everything we just learned about confounding, but the confounding examples were presented as to what to avoid when comparing treatments. Even though this integrated set wouldn't be suited for comparing treatment, it's more about finding rare events more easily versus our medical colleagues trying to add numbers in their head or manually across a whole lot of multiple tables. Typically, controlled integrated analysis sets are formed, and they tend to be the most useful integrated analysis sets that are used to compare treatment groups. So the general recommendation is to create these in a manner that avoids confounding of study and treatment, and then allows for the proper use of meta-analytical methods. I'll briefly go over meta-analytical methods in the next section. It is okay to combine studies of different lengths and populations within reason, assuming proper pooling and proper use of meta-analytical methods. Of note, when making decisions for what studies to pool, what you choose to pool for signal detection purposes, it does not need to match what will eventually be presented in regional labels. Integrated analysis sets created for signal detection purposes are those used to look for unexpected safety findings. In other words, look for the surprises. What is presented in labels is for risk communication purposes, and that can be different. They're often the same integrated sets, but it doesn't necessarily have to be that way. Additionally, what is pooled for signal detection purposes doesn't need to match what's done for specific safety topics of interest. One example is hypoglycemia in diabetes clinical programs. Many diabetes programs include studies with different background medications, and these different background medications might have very different safety profiles with respect to hypoglycemia. So for hypoglycemia, it may not be appropriate to broadly pool studies. However, that doesn't mean studies can't be pooled for signal detection purposes. Often the same integrated sets for signal detection are the same that are used for safety topics of interest, but it, again, doesn't have to be that way, and perhaps shouldn't be that way in some situations. In my experience, some submission teams think that the integrated analysis sets used for signal detection, risk communication, signal clarification for safety topics of interest all have to be the same. Well, that does lead to inefficiencies in team conversations and can lead to unnecessary volume. Having clarity on the context of these pooling conversations is very helpful. Let's go through an example to see how to apply these recommendations. I know this is a little bit hard to look at during a webinar, but we can go through it, and this might be something you might want to go back to later in looking at the recording and the slides. So let's look at this clinical program that has 6 studies. Study 1 is a phase 1 study in healthy volunteers. Study 2 is a phase 2 study in patients with a placebo arm and 3 dose arms. Study 3 is a phase 3 study with a placebo arm and 2 dose arms. Perhaps the low dose arm showed no efficacy, so it's kind of removed from consideration from later studies. It also includes an open label extension period. Study 4 is a phase 3 study similar to study 3, but doesn't have that open label extension period. Study 5 is an extension study. Study 2 and 4 patients can feed into it. So it doesn't have any new patients, it just has additional exposure from patients who participated in study 2 or 4. Study 6 is a phase 3 study without a placebo arm focused more on comparing middle dose and high dose. So let's think through what integrated analysis sets might make sense for signal detection purposes. One integrated analysis set to consider would be a placebo-controlled analysis set that includes studies 2, 3, and 4, which are the studies that have a placebo arm, pool the doses, and compare study drug with placebo. So when studies have different dose arms, pooling doses and comparing with placebo does provide a very nice, powerful analysis set for finding events that might warrant further scrutiny. Now, it couldn't be used to look for dose differences, but it could be very helpful in finding events potentially caused by the drug, but without regard to dose. Another integrated analysis set to consider is a placebo-controlled analysis set that includes study 2, 3, and 4, but don't pool the doses, and compare placebo, middle dose, and high dose. So all 3 studies have all those 3 treatment arms. It would leave the low dose arm out of the analysis. So the low dose would have to be assessed within the tables and figures created just for study 2, which is the only study that has the low dose. So now this program has a lot of data comparing middle dose and high dose, and likely of particular interest. So the most powerful assessment of middle dose versus high dose would come from combining studies 2, 3, 4, and 6. Because all 4 studies have both dose arms. Now, study 5 also includes both middle dose and high dose, but those patients aren't treatment naive. Only patients who tolerate study drug would even make it into that extension study. So including that study would create an underestimation of any treatment effect. So it's best excluded. Now, study 2 has substantially fewer patients relative to studies 3 and 4. The first analysis set may not add sufficient value. Now, assuming we'll use proper meta-analytical methods, we're not too concerned with combining the studies with treatment periods of varying lengths. In the big scheme, 12, 24, or 28 weeks aren't all that different. So what analysis sets are we not doing? Generally, we would not combine studies 2, 3, and 4 and compare placebo 5, 10, and 15. The placebo arm would come from 3 studies. The low dose arm would come from 1 study. The middle and high dose arms would come from 3 studies. So as we learned, that introduces confounding of study and treatment. Now, if the low dose arm is sufficiently large, there are meta-analytical approaches that can be considered so that low dose arm can be included in a dose relationship assessment. That's a Bayesian indirect method. I included a reference for this example of that method, but it's out of scope of this talk. In most cases, you can use direct comparison methods. Also, we don't have an analysis set that includes study 1, because generally we don't pool healthy volunteers with patients. Moving on to the next common mistake, oversimplifying safety summaries. We should try to make everything as simple as possible, but not simpler. Let's pause for another poll question. Do you know what Simpson's paradox is? No? Well, you've heard of it, but maybe you're not sure you fully understand it. Or yes, you've heard of it and you fully understand it. We'll pause for a few seconds. Okay, maybe just a few more seconds. Okay. So when deciding what studies to pool and what statistical methods to use, another concept to understand is Simpson's paradox. This is best understood through an example. So here's an example of Simpson's paradox. So within each study, the percentage of the event is higher with the treatment group than the placebo group. However, when you create Crewley pool percentages, 1.23% and 1.19% in this example, it appears there's no difference at all, which is why it's called a paradox. So even if you're aware enough to only pool studies that include the same treatment arms, crew pooling can still lead to misleading results. So now using a meta-analytical method that stratifies by study will help. Stratification by study is not a difficult concept. Basically, you calculate your treatment versus placebo effect within each study first, odds ratio in this example. You then take a weighted average of that effect. So in this example, if you create Crewley pool percentages across the studies first, then calculate the effect, you get 0.97 as the odds ratio, which is very misleading. It appears there's no difference. Now, if you first calculate the effect within each study first, then take a weighted average of the effect, you get 1.3 as the odds ratio, which is a way better representation of the data. So now, while stratification is a relatively simple concept, it's not yet broadly used across integrated clinical trial safety analyses. One purpose of this webinar is to change that. So stratification methods should be routine when pooling control data from multiple studies. This example is from the Advances in Collating and Using Trial Data ebook chapter, and that's in the references. So when a colleague of mine and I gave a similar presentation to a group several years ago, one member of the audience said, geez, with all these issues with pooling data, why do we ever pool studies? That was a very insightful comment. Not pooling studies at all is better than inappropriate pooling. However, we do think there is value to pooling studies appropriately to help with the overall effort of finding events that warrant attention. It could be easy to miss a signal if your medical group just has a large volume of by-study results. However, there could be some clinical programs in which keeping studies separate is best. Here are some general recommendations when deciding on the statistical methods. For controlled integrated analysis sets, use proper analytical methods. Meta-analytical methods. As mentioned previously, create your integrated analysis sets in a manner that allows you to use proper meta-analytical methods. So in most cases, that means pooling studies that have the same treatment arms that are being compared. Some people believe that a meta-analysis is when you combine data from literature. So combining data from multiple studies across a clinical program is also a meta-analysis, and meta-analytical methods should be typically used. Also, include the study size adjusted percentages. To learn about study size adjusted percentages, we do have this link to an educational video. It's only about 10 minutes long, so you can do that on your own time. But from a practical perspective, calculating stratified risk differences, risk ratio, and odds ratios should be routine. They're easily available in most software packages, and they take the same amount of space on a page as an unstratified difference or ratio. However, regarding study size adjusted percentages, there are some situations where it just may not be worth the effort or page space to include it. They are not yet easily available in most software packages, to our knowledge, and they would take a bit more space on the page, assuming most people want to keep those observed percentages. This educational video does outline such situations in which they're more helpful, which situations in which you kind of really do need them versus not. So here's an example of a forest plot for adverse events. This is from our 2017 PHUSE Adverse Event White Paper. When this plot is created for integrated control analysis set, the study size adjusted percentage and the risk difference stratified by study can easily be used. So they're very easy to include in a display. Here's an example of a forest plot for a safety topic of interest. The by study results can be included with the study size adjusted percentages and the risk difference stratified by study used for that pooled results row at the bottom. Here's an example of a display for a rare safety topic of interest. So this display shows the data by study. So for rare events, perhaps a pooled summary isn't all that helpful. So this is one way to display the data. For rare events, individual case reviews are usually what informs decisions on whether an event is reasonably likely caused by a study drug or not. But this is a very nice, useful way to kind of show where the events are occurring. And this is an example from our 2021 PHUSE Safety Topics of Interest white paper. When deciding on statistical methods, another important consideration is what metric or metrics to use. In general, there's a lack of awareness of the pros and cons of the various choices. All the metrics come with some assumptions, and these assumptions are not always well understood. In practice, there tends to be an over-reliance on the percentage. Relying on percentages is often okay, but not always. We do have a discussion of this topic in the 2017 PHUSE white paper. Now, some researchers might think we should be using exposure-adjusted incidence rates when there are differences in observation time between treatment arms, or when there are different lengths of the treatment periods across studies for a pooled analysis. There are some advantages to this approach, but there is a constant risk assumption that isn't always appreciated. When a study drug causes the event, it's not unusual that the event occurs early or late, thus not following that constant risk pattern. So we all need to do better on thinking through the appropriate metrics for any given study and clinical program. Different methods may even be needed for different safety topics of interest. So there is a section in the 2017 8-week white paper that briefly discusses these metrics, but really further research and education on this topic is needed. And perhaps more of this will be covered in a future webinar. Another common mistake is to create unnecessary volume for safety analyses. While some might think there's nothing wrong with a large volume of safety analyses, there are very reasons why volume should be right-sized. Time to create and view tables and figures that don't add sufficient value takes time away from those tables and figures that do add value. It's more difficult to ensure quality if you're creating thousands of tables and figures versus creating 100 of well thought out tables and figures. So creating unnecessary volume adds time, and the patient is waiting. So when creating integrated analysis sets, one way to reduce unnecessary volume is to avoid having two analysis sets that include almost the same set of studies and patients. So when deciding on the planned analyses, usually fewer analyses are needed for uncontrolled integrated sets versus controlled integrated sets. Teams should simply not take the list of planned analyses for one set and apply that same list to all integrated sets without some thought. Additionally, there are some commonly produced analyses that add little value. For example, as discussed in our 2017 PHUSE Adverse Event White Paper and several other documents, for clinical programs with controlled data, summaries of events considered related to the investigator adds little value. Several of our PHUSE white papers are intended to help researchers on right-sizing volume for clinical trial safety analyses, and I encourage you to read them. The last common mistake I'll cover is the overuse of past methods. We understand that it's difficult to do something different than what has been done in the past or what everyone else is doing. There's a tendency to stay with what has worked in the past for getting a new drug approved. However, that has become a barrier to improving our safety analyses. We need to find a path forward to improve safety analytics without jeopardizing the review and approval of a new drug. So collaborations such as the FDA PHUSE, public-private partnership, and other partnerships are intended to help with this journey. If you want to learn more about safety analytics, we encourage you to visit the PHUSE Safety Analytics education page. The site includes a lot of useful references. It's intended to be a nice place to start for those new to clinical trial safety, but it's likely useful even to those who are experienced. So I hope this talk brought awareness to how much scientific expertise is really needed to properly analyze clinical trial safety data. And I hope it has inspired you to learn a bit more. So thank you, and we'll pass it on over to Veronica. Sorry about that. Before passing it on to Veronica, we do have one challenge question. This, again, is something we do as part of offering continuing education credit. So here's the challenge question associated with this talk. So when combining data from multiple randomized controlled trials within a clinical program, it is considered a meta-analysis, and meta-analytical methods should typically be used. True or false? All right, well, I hope everybody got it right, because if you didn't get it right, then I didn't do a good job presenting. So yes, the correct answer is true. So thank you, and now we'll move it over to Veronica. Thanks.
Hello, everyone. Thank you, Mary, for that great presentation. I will now follow with a brief presentation on FDA Type C meetings on ISS safety analysis strategy and related data requirements. Here's a list of learning objectives for this session. At the end of the presentation, you will understand the different types of formal meetings with FDA. You will be able to describe how Type C ISS meetings can be used to support drug development programs. And finally, we will cover how to request a Type C ISS meeting. Let's start by talking about formal meetings with the FDA. Under PDUFA, there are 6 types of formal meetings between the sponsor investigator and FDA. The purpose of these meetings are to allow FDA to provide advice to sponsors throughout the drug development lifecycle. These meetings often represent critical points in development and hence follow timely and consistent procedures. The 6 different types of formal meetings with FDA include Type A, Type B, a subset of Type B referred to as end of phase, Type C, Type D, and Interact, which stands for Initial Targeted Engagement for Regulatory Advice on CDER and CBER products. Details on each of the meeting types are discussed in the FDA draft guidance on formal meetings, and I've provided the link at the bottom of the slide below in case you have interest to read about these meetings further. Let's now talk about each type of formal meetings with FDA. So Type A meetings are those necessary for an otherwise stalled product development. These meetings may include those to discuss clinical holds and dispute resolutions. Type B meetings we're very familiar with, and they include pre-IND, pre-NDA, or BLA meetings. The end of phase meetings, such as end of phase 2, fall into a separate category, but I've combined them here because they're all considered milestone meetings, which occur at critical junctures in drug development. Now, Type B meetings are also used to discuss overall development program for specific products that are granted special designation status, such as breakthrough therapy. Now, we're going to skip Type C meetings for a second, and we'll come back to it. The Type D meetings are focused topic meetings for discussion on a narrow set of issues. And there's usually a limit of 1 to 2 topics with no more than 3 to 5 questions. And the issues should not require input from more than 3 disciplines or divisions. Otherwise, the meeting is too complex and a Type C meeting should be requested. Now, an example of this would be when a sponsor has a specific question about an aspect of an innovative trial design or a specific question about presenting data, follow a pre-NDA meeting. Now, the Interact meetings are intended for novel products and development programs that present unique challenges early in development, such as before the filing of an IND or before having a pre-IND meeting. These issues discussed typically relate to IND requirements, and Interact meetings are intended to facilitate IND enabling efforts when the sponsor is facing a novel, challenging issue that might otherwise delay progress of the product toward entry into clinical trials. So coming back to the Type C meetings, basically, these are any meetings that does not fit in any of the other categories that I already discussed here. So let's now focus on the Type C meetings on ISS safety analysis strategy and related data requirements. This is a mouthful, so I will be referring to these meetings as Type C ISS meetings from here on. Now, ideally, the optimal timing for these Type C ISS meetings should occur approximately 1 year before the pre-NDA meeting. Typically, at that time, sponsors have started drafting a statistical analysis plan for the ISS, but really haven't started any programming work for pooled or safety analysis. And this is a good time to get input from the agency because there's still time to make modifications based on advice from the agency. Now, the goal of these Type C ISS meetings is to discuss safety analysis strategy for the ISS and any related data requirements. So let's now talk a bit on the types of topics that might be appropriate for discussion at the Type C ISS meetings. Now, we have heard from Mary that one of the common mistakes sponsors make is not seeking FDA input on the approach to aggregate safety analysis early enough. And now you can see that these Type C ISS meetings can be used exactly for that purpose. Sponsors can seek input from the agency on a number of different topics, including data pooling for safety analysis, analytic methods, approach to AESIs. Sponsors may also seek advice on related data requirement topics, such as data standards and submission requirements for data sets. Here are some example sponsor questions that could be discussed at the Type C ISS meetings. Does the agency agree with the proposed pooling strategy? Does the agency agree with the proposed integrated safety analysis plan? Does the agency agree with the proposed grouping of measure-preferred terms for evaluation of, and you would insert the appropriate potential safety signal that you're concerned about, or an AESI? Another question will be, does the agency agree with the proposed study data standardization plan? Are legacy data acceptable if SDTM and ATOM datasets are not available? And finally, does the agency agree with the proposal for narratives and case report forms? So to optimize the output of this meeting, sponsors should submit the following documents for review as part of the briefing package. A description of all trials to be included in the ISS, presented in a tabular listing of clinical trials. A draft ISS statistical analysis plan, and this should include a discussion on pooling strategy, rationale for inclusion of or exclusion of trials, any planned analytic strategy to manage differences in trial designs. For any phase 3 program that include trials with multiple periods, such as you may have a double-blind randomized period, a long-term extension period, sponsors should outline the method of assignment of study events to a specific period. Sponsors may also provide a list of previously observed and anticipated safety issues to be evaluated, and their planned analytic strategy, including use of any SMQs or FMQs, modifications to specific SMQs, or sponsor-created groupings of preferred terms. A rationale supporting their approach should be provided. Now, to request a Type C ISS meeting, sponsors should include the following phrase in the meeting request cover letter. Discuss safety analysis strategy for the ISS. Now, this phrase will alert the regulatory project manager to the specific meeting type. For Type C meetings, sponsors may request any of these following formats, including in-person, virtual, teleconference, or written response only, also known as WRO. However, the agency may determine that the WRO is appropriate, and the agency makes the final determination on the best format for the meeting type. Now, as part of the meeting submission, sponsor may also submit sample standardized data sets for evaluation. When doing so, sponsors should include the phrase in the submission cover letter, clinical safety sample data sets. Submitted sample data sets will be reviewed for conformance to data standards, structure, and format. Now note, these sample data sets will not be reviewed as part of marketing application. So many of you have encountered situations where these topics are discussed during the pre-NDA meeting. So you might be wondering why you should leverage the Type C ISS meetings. Well, there are a number of advantages. So the first advantage is that it is an extra opportunity to interact with the review division and seek agency guidance on an important topic that will impact the marketing application review well before the pre-NDA meeting. Now, as you heard from Mary's talk, planning for aggregate analysis should be started early, and you want to seek review division agreement on the best approach to pooling. Early interaction with the agency will also allow sponsors adequate time to implement any recommended changes to the safety analysis approach without delaying the market application submission. And finally, as we know, this is an important topic, so we really don't want this topic to be lost in the large number of questions typically needed to be discussed at the pre-NDA sessions, which is why we think a separate meeting is warranted. So in summary, now that you have learned all about the Type C ISS meetings, I hope that you will leverage these meetings to engage in early discussion with the agency on approach to aggregate safety analysis. Now remember, the best time to request these meetings are after you've initiated your phase 3 studies, but before the pre-NDA meeting. And typically this is a year before the pre-NDA meeting that you want to consider requesting these meetings, but after you have drafted the ISS statistical analysis plan. Now this will still give you plenty of time to implement any recommendations made by the agency. So now we have a challenge question. Which of the following statements regarding Type C ISS meeting is not true? A, they are optional. B, can be conducted via a written request only, in person, or teleconference. C, best conducted before the pre-NDA meeting. Or D, best conducted before drafting the analysis plan for the ISS. Now take a second to consider which answer you think is correct. And if you pick D, you are correct. We discussed that the best timing to request a Type C ISS meeting is after you have drafted the analysis plan for the ISS. So here are the list of resources for your reference. And this concludes my presentation, and I will now invite Dr. Greg Levin from the Office of Biostatistics for some brief remarks. Thank you.
Well, thank you, Veronica, and thanks for the opportunity to comment today. So I enjoyed the presentations by Mary and Veronica and agree very much with the principles and messages that were conveyed. I'll just take a few minutes to reinforce a couple key points. First, I want to underscore the importance of quality upfront planning in the evaluation of drug safety. This includes utilizing the Type C meetings on integrated safety analyses, which can be helpful to align on an analysis plan for the integrated summary of safety well in advance of an NDA submission, as Veronica outlined. But I think that quality planning for the safety evaluation also involves appropriate choices for elements of the design and the conduct of clinical trials. And such design and conduct choices need to be proposed and discussed with FDA at earlier meetings in drug development. So, for example, end of phase 2 meetings. I'll share a few examples of the kind of planning I'm thinking about. So suppose that non-clinical findings or early phase data or data from other drugs in the same class generate some concerns about a potential serious drug risk that's classified as an adverse event of special interest. As part of the planning of the phase 3 clinical trials, there should be careful consideration and discussion about how data on that adverse event will be ascertained, including whether the trials will rely on routine open-ended adverse event reporting or will include some targeted data collection to ensure reliable and comprehensive data are collected on the specific potential risk. There should also be a clear case definition of what will constitute an event in the trial and in analyses, as well as consideration about how to prevent missing data in important analyses, including approaches to continue to follow and ascertain outcomes in participants who discontinue treatment early. And finally, depending on the clinical context, there may be value in sample size calculations to understand the degree of precision that the program will provide on the adverse event of special interest and whether that is sufficient to inform the benefit risk assessment. These aspects of quality planning for the evaluation of safety are very important and should be considered and discussed as early as possible and prior to initiation of phase 3 trials. The second point I'd like to emphasize is the importance of using appropriate statistical methods given the trial design and the underlying scientific question of interest. To provide a few examples, I can just reinforce some of the messages that you heard in Mary's talk. To avoid the potential for bias, safety analyses that integrate data from multiple trials to compare two treatment arms, for example, let's say a proposed dose of a drug and placebo, should generally involve only those trials that included both treatment arms. In addition, such integrated analyses should always stratify by trial. There should also be careful consideration about the choice of statistical metric. In many cases, the use of crude proportions or percentages will not be appropriate, and it'll be better to use an alternative approach such as Kaplan-Meier estimates or incidence rates that account for differences between participants in at-risk time. This would be the case in common scenarios such as where there are event-driven trials in which participants are followed for different durations, or where the integrated analyses include trials with different durations, or where there's considerable participant dropout. So I'd really agree with, I think, what Mary called common mistake number 6, that an important barrier to improving safety analytics is assuming that what everyone has done or is doing is the best way to go forward. We really should think carefully about the most appropriate statistical methods for the setting and then utilize those methods to ensure reliable safety results and decision making. So those are just a couple quick comments. I'll stop there and just reiterate that I enjoyed the 2 talks and I look forward to further discussion in the Q&A.
All right. Thank you again to our presenters and speakers for their excellent presentations and insights. We will now begin our question and answer panel. If you haven't had a chance to enter your question into the Q&A chat pod now, please do so now. We'll answer as many questions as time allows. All right, and now our first question will go to Mary. Mary, can you elaborate on how we may find rare events when we integrate an all-study drug integrations? Is it simply because we are increasing our overall big N or exposed population?
Hi. Yeah, that's a good question, and I will expand on what the point I was trying to make. So when you create an all-study drug integrated analysis set, it will allow you to find those rare events a little bit more easier, just because if you don't have that integrated analysis set, your medical staff will just need to try to find those by looking at multiple tables, and it might be harder to find those. So when you have a single table that says, here's how many patients had a particular event across our entire clinical program, it just might be easier to find those, really. I mean, so it does perhaps give you a better estimate of your percentage or your exposure adjusted incidence and so forth, but really it just makes it easier to find those events that might just be harder to see when it's scattered across multiple tables. And then you can see how many patients had the event, and you might be most likely for these rare series events want to follow that up with finding those patients and reviewing their specific cases, for looking and do a medical review of all the individual cases and look for things like re-challenge, de-challenge information. Are there alternative causes? Were they taking certain medications? So just makes it easier to find those events that might deserve attention and then require some follow-up with medical case reviews. Thank you. Hopefully that answers the question.
Great, thank you for that response. One more question for you, Mary. When you describe population, does it mean population with same indication?
So when I was referring to populations, when I said things like you can pool data across various populations as long as you use the correct meta-analytical methods, I was using that a little bit broadly. I would think for the clinical, most situations, that does mean kind of across indications, because that's what we mostly see is a molecule being studied for different indications. But it could mean other populations too, like maybe different age groups or different regions, different other things. So there could be different ways in which you might have a different population. But I think in the context of that word in my presentation, it really was kind of meaning different indications.
Okay, great. One more question for you. Is it worth calculating study size adjusted percentages if you have treatment columns that you have no intention of comparing? Say they are side by side for ease of viewing, but no comparison of treatments is of interest, i.e. no risk ratios or differences calculated?
No, you would. Yeah, thanks for that question. So I'll try to clarify it a bit more. So study size adjusted percentages are truly only warranted when you're trying to compare treatments. And your Mantel-Hansel risk differences or Mantel-Hansel odds ratio stratified by study, those type of metrics are useful when you're comparing treatments. So all of that is in the context of comparing treatments. However, I would question why you would have a table with different treatment arms when your goal isn't to compare treatments. So if your goal is to compare treatments, I'm not sure why you would have a table with all these different treatment arms. So maybe it is, I would try to pick a different display maybe, because if you have a table or figure with different treatment arms, the temptation is going to be to compare them. And if it's not appropriate to compare them with that display, then I would question why you would even have that particular display. Maybe there's a reason, and I don't know the context of why you might have such a display. But it is a common problem to create displays that makes it really easy to do poor comparisons. So I would try to avoid creating displays that makes it easy to do poor comparisons. So I would think about whether you should be doing that table to begin with, and if whatever you're trying to accomplish with that table, maybe there's a better way to do it. So thank you. Yep.
Okay, thank you again for that response, Mary. And now we'll move on to questions for Veronica. Veronica, first question, can you clarify the difference between an SCS and ISS?
Yes, thank you for that question. So the SDS, or the Summary of Clinical Safety, versus the ISS, or the Integrated Summary of Safety. All right, so these are two separate documents, if you will, that will be submitted to two different sections of the ECTD. So the Summary of Clinical Safety it's typically a text content, and it's just basically a word summary of the clinical safety. Is intended to be an overview, a high-level summary, very brief. It's intended to be submitted into Module 2, and there is a space limitation of 400 pages in the ECTD system. Now, the ISS is sort of misleading because the name sounds like it's sort of a summary, but it's intended to be much more than that. It's not really a summary. It's intended to contain detailed integrated analysis in addition to a more detailed explanation of the summary. It's intended to include also the integrated analysis, all relevant data. That includes tables and listings and data sets. And there is no space limitation. And that's intended to be submitted to Module 5. Now, if you read the FDA guidance, which I'm not sure if we can share the link to the guidance, but there is actually an FDA guidance that talks about the submission of the ISS. In rare instances, it may be acceptable to just submit the narrative portion of the ISS in Module 2 because they're often very similar, especially in cases where the application is very small or it's a very single study with very limited data. But before the sponsor decides to, and the ISS is required by regulation, so the sponsor should meet with the review division and discuss during the Type C meeting if you feel like your application meets one of those rare instances where an exception can be made. And if you look at the guidance, it also includes a lot of examples, several case examples on how you should submit the SCS versus the ISS. So I highly suggest that you look at that guidance document.
Right, thank you for that response. Another question for you, Veronica. Why are healthy volunteers not recommended in the all-study drug pool?
So I think the answer to that is it's not to say that they're not all recommended. If you look at a marketing application submission for approval, typically we're reviewing safety data relevant for the intended to be marketed dose in the intended therapeutic population for that indication. So healthy volunteers really don't meet that criteria as far as the targeted population who will be taking this drug. And also there are a different study population because the population with the disease may have some predispositions that place them at a higher risk for certain adverse events. So in general, the safety analysis is focused on the targeted population for the intended therapeutic indication. And so I wouldn't say they're not recommended. Certainly in some cases, you may look at all populations that has received at least one dose of the drug, especially in cases where there's rare disease or there's very, very few population. So you want to look at everybody possible. But in general, if you're looking at just the best data to look at the safety summary is in the intended population.
Okay, thank you for that response. Another question for you, Veronica. Are the sample standardized data sets the proposed integrated data sets or a single study data set? And just checking with FDA, it's acceptable?
Yeah, so basically, again, there is a designated site that provides information for a sample standardized data set. And I think I did include that in my in my slides, but if not, you can easily Google that. So there's no real requirement for the type of data set that you're going to submit. You can choose to use an integrated data set or a single study data set. The intention of the sample data set submission, it's primarily for those sponsors who's never ever submitted a data set through the ECTD, and the intention is so that it does all the checks for the standardization to make sure that the submission will go through. So there's no real requirement how you lay out the data. And if you have more questions, you can also reach out to the eData team at the FDA, who are going to be those that are responsible for reviewing the sample data sets.
All right, thank you. And one more question for you. Are we obliged to use the FMQ, or is it acceptable to use SMQ?
Yeah, so that's a great question, and we get that question a lot since that we released the FDA Metro Queries public. And there is no regulatory requirement to use FMQ in your safety analysis. This was intended just for FDA to be transparent in the process that we've developed in our safety analysis. Sponsors are welcome to provide even custom queries or also modify the MEDRA SMQs as long as you provide a rationale in your submission. So this is a perfect type of questions to discuss at the Type C ISS meeting, where if you intend to propose a set of preferred terms that you want to customize for a specific potential safety signal evaluation, this is the perfect time to seek division input on the appropriateness of the terms that you intend to include.
Great, thank you again for that response. Now we'll move on to a question that may be addressed to Greg. If AP2 and PH3 studies are similar in design and population, but differ in risk mitigation measures for a safety event of interest, would you recommend pooling for overall safety and then separating for purposes of characterizing the EOI?
Thanks for the question. This is a good question, and I think it risk differences in risk mitigation measures between trials kind of falls in that area that I think Mary alluded to, where that could qualify as like a important difference in design and conduct that could modify treatment effects, for example, on specific safety events of interest. So, I guess my short answer would be it's probably going to depend on the nature of the risk mitigation measures and how much of an impact that they are likely to have on the treatment effect on that specific event of interest. And there probably is going to be value in looking at results from integrated analyses and results from individual trials, for example, to be able to see whether the risk mitigation measures made a difference, and to see whether and to estimate effectively the magnitude of the risk in the presence of the risk mitigation measures, particularly if they're likely to be implemented in clinical practice if the drug would be approved. So probably there's going to be value in looking at it both ways.
Thank you for that response. I've got another question for you, Greg. Should the stratified analysis, such as CMH odds ratio, only be done for events with a specific interest? It's not practical to provide such adjusted analysis for all events.
Thanks for the question. My recommendation would be that stratified analyses of so when there are integrated analyses of data from multiple trials, that analyses that stratify by trial are done for all events. This is a pretty straightforward statistical calculation. Doesn't have to be a Cochran-Mantel-Hansel odds ratio. There are things like study size adjusted percentages that are, essentially one additional line of code in calculating and estimating the percentages within treatment arms and in estimating a comparison between the treatment arms. So from a from a practical standpoint, in terms of the underlying programming, it is very feasible, and for the reasons that Mary outlined in her talk, prevents the possibility of bias in the extreme case Simpson's paradox. So is important to ensure that we get reliable results, not just for adverse events of special interest, but additionally for signal detection as well. So I would recommend it as a general approach for all safety events.
Right. Thank you for that response. And now we'll go back to Mary for some additional questions. Mary, should we be pooling studies across indications?
Yeah, so thanks for that. And that's a very frequently asked question and relates to some of the other questions we received. So yes, I do think that if you have placebo-controlled studies in multiple indications, so let's say you're studying a particular molecule across multiple indications, I do think an integrated analysis set that includes the placebo-controlled studies from all those various indications can actually be very helpful. Now, does that actually get submitted as part of a submission for a particular indication? Maybe, maybe not. That might depend on various things. But for your own sponsor's program level safety review, I think those kind of integrated analysis sets can actually be extremely helpful in finding events that warrant your attention. And then once you find those, then maybe you could do additional summaries to kind of look into it to see if you really think it's reasonably likely caused by the drug. But that could be a very powerful integrated analysis sets to help you find the things that deserve your attention. But yeah, that doesn't always get included in a submission if that submission is for just one of the indications. The exception might be if, let's say, you're submitting for one indication and you have all these other data for other indications, and your submission will probably focus just on the indication that you're trying to submit. However, if in your analyses you found something that only that pool group kind of brought to your attention, or when you try to decide what should be in a label, and that other data is important to be part of your label, well, then you would need to perhaps put some of those analyses in an appendix, because if you do propose something in labeling, that you would need to kind of annotate it too. So if those additional analyses kind of uncover important information, that would probably need to make its way into your communication with regulators. But in general, it's very useful for your own program level safety reviews and can be very helpful. I can pause to see if Greg or Veronica wants to add anything to that question. Okay, they're not answering. I do, just to add to one more thing, I think in my slide, I would kind of use this word within reason. I am aware of one example where a molecule was studied for a cancer indication and another indication was for an autoimmune disease, right? That might just be too different, right? I guess when it's helpful to provide data and combine indications, we're not talking about some of those extreme examples where the type of events a cancer patient might have is just so different than the type of events that an autoimmune population might have. Some of that just could be too different. So there is some judgment applied, but I do think people are a little too nervous and think that the indications have to be almost identical or super close to be able to find value in pooling them. I think some degree we're a little too cautious. But yeah, there are cases where if they're just too different, I would never put them together. So hopefully that addresses that a little bit more. Thank you.
Okay, thank you, Mary, for that response. We'll do one more question for you. Is it ever okay to compare placebo data from controlled trials with data from open label trials?
Yeah, that's another frequently asked question. So yes, thanks, because it is certainly pertinent to talk about this. So it happens in real clinical programs is we do have a lot of data often of uncontrolled data, open label extension data, open label extension periods of studies. And interesting enough, those are usually included in study to gather more safety data. But what happens is we get safety data from all that uncontrolled, and we don't know what to do with it. We don't have anything to compare it with. So a lot of times we have that data to really give more opportunity and more exposure to see if there's any kind of rare series of things that might come up. And so really we should be kind of considering that data in that context. So I don't think in general we shouldn't necessarily feel the need to compare that with placebo-controlled data from placebo-controlled trials. That being said, I think people still might want to kind of look at it, put the data in context. I don't think it's ever appropriate to statistically compare it, right? To make formal statistical comparisons with open label data with a placebo-controlled arm of a study, right? That you have to treat that like observational data. It's very similar to observational data as if you're comparing with literature. It's not much different than that. So I would say, no, you shouldn't really be officially comparing it, but in reality, some people might look at it to kind of put some of that open label data into context, but there could be different data that you should be looking at to put that into context, like literature-based background rates or some other type that you might have. So it really does fall into some medical judgment, but I wouldn't overly compare uncontrolled data from uncontrolled studies or phases with placebo data. You have to know what you're looking at and know the differences between the data you're looking at and treat it like observational data. Thank you.
All right, thank you for that response. Looks like that is all the questions we have time for. Thank you again to our presenters for all the excellent information.